Respiration is essential for all living organisms. And so we are going to talk about the basic concepts first, and then we will look at the individual processes that happen in respiration. Okay. So this is what keeps all the cells going by producing enough ATP. And it happens in multiple ways. Most of these processes are aerobic processes with the presence of oxygen, but there are also anaerobic processes that in the absence of oxygen. So in the aerobic processes, the whole thing is divided into multiple steps, such as glycolysis, acetyl-CoA formation, then Krebs cycle, and oxidative phosphorylation. And there are processes that are anaerobic without air or without oxygen involved. And so this involves anaerobic respiration and anaerobic fermentation. Okay. So the anaerobic respiration and anaerobic fermentation, they are two separate processes. So the fermentation is what we will look at as the alcohol fermentation and the lactate fermentation. These are the two separate processes. So we will see, where we get the energy from the food that we consume, and how that energy is harvested and how that energy is used. All that is what we are going to see now. So in a cell, we need to also understand where these things are happening. So the place in bacteria and in eukaryotes. Okay. So in bacteria, all these things happen in cytoplasm, starting from glycolysis to Krebs cycle. And the oxidative phosphorylation happens on the plasma membrane or cell membrane. Okay. And these two happen in cytoplasm also. So in the eukaryotes, the glycolysis happens in cytoplasm, and the acetyl-CoA formation and the Krebs cycle happen in mitochondrial matrix, and oxidative phosphorylation happens on the inner membrane of mitochondrion. And the fermentation happens in cytoplasm. So the structure relates to function, applies to all these processes, and there are some special structural features of mitochondrion that allows this to happen. Okay. So the basic concepts is first thing that we are going to look at. Okay. So the basic concepts, so the energy stored in food in the form of carbohydrates. This can be simple carbohydrates such as monosaccharides or disaccharides, or complex carbohydrates such as starch or glycogen. Lipids, mainly fats, and proteins. So these are the main sources of energy. Okay. Are harvested in this process, okay, to make ATP. And recycle organic compounds. Okay. So another thing in this process that happens is that after you extract the energy, not all of the molecules will continue. They are used to build the body, and that's something to understand. Okay. So the prime example of respiration, this is expressed in this summary equation. So the summary equation goes with glucose, C6H12O6. That's the most common form of energy that we get from the food we eat. So that is combined with 6 oxygen, and that releases 6 carbon dioxide plus 6 water plus energy to make ATP. So that is the summary equation, but it doesn't happen in one step. It happens in multiple steps, and the energy is extracted from the food and used to make ATP. Okay. So we need to think about where is this energy coming from. Okay, so all these are, coming from the covalent bonds, and we are talking about electrons spinning in higher orbits or lower orbits. So when the electrons drop from high to low orbits, that releases energy. And if they go from low to high orbit, they consume energy. So that's where the energy is coming in. And the energy is stored in the form of covalent bonds. Okay, so that's one thing. When you talk about covalent bonds, it is nothing but sharing a pair of electrons. So these are electrons going from low to high orbits. So that's where the energy is going to come from. Okay. So energy is stored in electrons. Okay. So then we need to think about what happens. We gain electron, lose electron, and so on. So we have to think about this glucose with C6H12O6 becoming CO2 has lost that hydrogen. That process of losing hydrogen is called as oxidation. Okay, is loss of electrons and our hydrogen. Okay, so hydrogen is nothing but one proton, one electron. and the proton is referred to as a H plus ion. Okay. Oxygen, on the other hand, becoming water molecule, has gained hydrogen. That is referred to as a reduction. Okay. Reduction is a gain of electrons and or hydrogen. So the way you can remember this is by thinking about oxidation is a loss and reduction is gain. So the acronym is oil rig. Oxidation is loss, reduction is gain of electrons or hydrogen. So this whole process of respiration is an oxidative process. Okay. Is an oxidative and catabolic. Okay. It's an exergonic, okay, and exothermic process, okay, that increases the randomness. So this kind of summarizes the nature of respiration. So the next thing to look at is what carries those electrons. So electrons, like electricity, you don't want to let that just freely float in the cell. So they have to be carried. So there are specific chemicals called as electron carriers. They are also called as the organic cofactors. So these are molecules that accept and donate electrons, and they are considered as cofactors. Okay? So the two major electron carriers used in respiration are NAD+, which stands for nicotinamide. Nicotine is a type of nitrogenous base. Adenine, that's another purine nitrogenous base we saw in ATP dinucleotide. And this one has a natural positive charge. That's how it will be denoted as NAD+. Okay. So this NAD+ can accept 2 electron from 2 hydrogen, and it becomes, it can go both ways, becomes NADH plus a proton. Okay, so you have to understand how it carries 2 electrons. So the 1 electron neutralizes the positive charge, and another electron and a proton goes in to become this hydrogen, and other proton is released. That's how the 2 electrons and 2 protons of this H2 are carried. Okay. So the other type of electron carrier used in respiration is called FAD, which stands for flavoadenine. This is flavin is another nitrogenous base, dinucleotide. So these nucleotides are the one that can accept electrons. So the way FAD accepts is it takes 2 electron and 2 proton, and it becomes FADH2. That's how they will combine. Okay. So when they gain electron and hydrogen, as you know, this process is called as reduction, and this process is called oxidation. It's the same thing here. This process is reduction, and this process is oxidation. Okay. Any questions so far?
I have one.
Yeah, go ahead.
So why is it that it's not FADH plus H like the top one?
You're talking about this equation?
After reduction, how it's all together and not like the NAD where it's NADH plus an extra H.
Yeah. This is a proton. Each hydrogen contains one electron and one proton. So 2 hydrogen atoms contain 2 electrons, 2 protons. 1 electron goes to neutralize this positive charge, so there is no charge on NADH. Another electron and another proton become this hydrogen, and a proton comes out. So you're accounting for 2 protons and 2 electrons coming from this.
Okay, so it's because the FAD isn't a positive charge?
The FAD doesn't have a charge.
Okay, I see.
So it accepts both the electron and the proton become FADH2.
Okay, cool.
Good point. Any other question? So it's interesting, we don't realize all the power systems we have run on electricity. So the source of electricity can be different. It can be coal-fired or gas-fired power plants, and solar, wind, hydro, and different sources, but the electricity is the same. Electron flowing through from, one electrode to another electrode. In this case also, our body and our cells depend on the power of the electrons. The amount or the voltage is different. We are in millivolt compared to 110, 120, or 240 volts. So that is how we need these electron carriers to survive. Okay. And the process of respiration is so important that you can live without food or water for a week or 10 days, but you cannot live without oxygen for more than 3 minutes. It's that important. And why? Our cells make, each of our actively working cell makes and uses about 10 million ATPs per second. Imagine that. So you have to constantly make that ATP, and we will talk about it, when we discuss it in this core cap. So I'm going to continue this as to how do we use this to make ATP. So try to make that connection. So ATP synthesis is the next we will talk about. How does that happen? Okay. So the ATP synthesis happens in 2 ways. One, by substrate level phosphorylation. As you know, phosphorylation means adding phosphate. And so here you take the phosphate from a chemical X and you give it to ADP, and you make the ATP, and the X comes off. Okay. It's a very single step process, and the generic name of the enzyme that does this called kinase. Okay. Generic name, kinase. Adds a phosphate to ADP or any other molecule, and so this is a high energy phosphate molecule, And the example of this, some of you may have heard of the chemical creatine. Do you know where you heard of the creatine? Anyone use
Energy
creatine?
drinks,
Say that again?
energy drinks and like protein.
Yeah, energy drinks. You would find people also take that tablets as a supplement during exercise, but their creatine is nothing but creatine monophosphate. So when the cells are running out of oxygen during exercise, so you pop this pill in, this is a stable form of high energy phosphate. It combines with ADP and it releases ATP and the creatine. It comes out without the phosphate. So that is how creatine helps you keep working out when the cells are running out of oxygen. So that's a benefit of the creatine. This is a one-stop process. The enzyme kinase does it. Just take the phosphate from one chemical and give it to ADP to make ATP. Okay, but this is not a very efficient process, so it cannot run for a long time. You will find that in glycolysis. That's a substrate level phosphorylation. It makes a couple of ATP. And Krebs cycle, this happens. And fermentation. So fermentation is nothing but glycolysis and some byproduct formation. We will see that later on. Okay. So the major process by which ATP is made is called oxidative phosphorylation. Oxidative phosphorylation. So this involves the process of oxidation that results in phosphorylation. So that happens in the plasma membrane of bacteria, as we saw before. And on the inner membrane of mitochondrium. And I'm going to go through in detail roughly, and we will come back to it again in the eukaryotes. Okay. So here, the structure of mitochondrion is what I'm going to draw. So if this is the mitochondrion, this is the outer membrane. And the inner membrane, as you know, is kind of folded like this to increase the surface area. That's the inner membrane. And in between the two is called as intermembrane space. Okay. So the space within the inner membrane is referred to as the matrix. And these type of foldings you find here, all these projections like foldings, they're called as crista or cristae. So plural. All right, what happens here now? We need to connect the electron carriers and ATP synthesis. Okay, let's see what happens in these cases. And let's look at just one of the electron carriers. And they are going to give electrons to, there are protein complexes on the inner membrane. And so these protein complexes are called electron transport chain. or ETC. This is a short abbreviation, electron transport chain. It's like a series of proteins located on the inner membrane. I'm just going to put a few here and there so you get the picture. Okay. So in addition to that, the inner membrane also has proteins that can make ATP. Okay. So those proteins make ATP called ATP synthase. So this is like this mushroom-shaped protein that makes ATP is facing the matrix. Okay, so it's facing the matrix in multiple places. This is just some sample that I'm drawing. So this is ATP synthase. Okay. All right, so far so good. Any questions up to this point? All right, when the NADH now carries the electron, 2 electrons and the 2 protons, it is made in the glycolysis, acetyl-CoA formation, and the Krebs cycle. It comes into this electron transport chain, and it gives off the electrons and protons, and it becomes NAD+. So during this time, 2 electrons will go through this electron transport chain, and the 2 electrons, I think it's a negative sign, plus 2 protons that comes out of this NADH. Another proton is picked up from here, plus half of oxygen that we breathe in. All these will combine to make water molecule as the byproduct. That's where water is found. Okay. But that's not the end of the story or end of electrons. While this is happening, protons are being pumped. Okay, the protons will go through this electron transport chain, and they get concentrated in the intermembrane space. So you have a high proton concentration in the intermembrane space and a low proton concentration in the matrix. Okay, so all these protons come back and they get concentrated here. So let's put a lot more protons here, some protons in this case. Okay. So the intermembrane space has high proton concentration, and I'm going to put a square bracket here, and the matrix inside here has a low proton concentration. So, okay, so you're just creating your proton concentration gradient. And then what happens? So these protons now have to come back through the enzyme ATP synthase. When they come back, they create a force, just like water running through a turbine, and that helps in combining ADP and PI, inorganic phosphate, to make ATP. That is where the phosphorylation happens. Okay? So the protons, that happens in every ATP synthase. I just wrote in one place, you know, how the electron transfer happens. So how the overall thing is connected is something to understand. And I'm going to connect the dots now. So conversion of NADH to NAD plus, giving off these electrons. So this process is called oxidation of NADH to NAD plus. Okay. So that oxidation is what allows the protons to be pumped in. This happens in all the electron transport chain. That leads to generation of a proton concentration gradient. Okay. That's called delta proton concentration gradient. That leads to a process of protons coming from high concentration to low concentration called chemiosmosis. And chemiosmosis is nothing but protons going from high to low concentration. It's a chemical osmosis, low concentration. Okay. That leads to ATP synthesis, and this is nothing but phosphorylation of ADP. Phosphorylation of ADP. That's what completes this whole process. So oxidation resulting in phosphorylation is what is called oxidative phosphorylation. Okay. So here there is a force called as proton motive force. That is what drives this process, proton motive force, that makes the ATP synthase works, turn like a turbine, and allow that to make ATP. Okay. Most of the ATP we make is through this process, for which oxygen is essential, and oxygen is the final electron acceptor, without which this process will not happen. Okay, and we are going to see the details of all these later on in more in depth. So what I have just introduced to you is, what is the source of energy in the form of food? That's covalent bonds and shared electrons, and there are electron carriers that carry that electron and bring it to this electron transport chain, and that allows proton concentration gradient to form. That proton come back through ATP synthase, makes ATP. So that is the connection that you need to make. And oxygen is essential for accepting the electrons at the end, so that the whole electron flow can happen. Okay, so that completes the lecture for now.